Alrighty, guys, good morning and welcome to part 2 of the DNA replication lecture. We talked yesterday about the actual process of replication and how it's called semi-conservative replication. So you end up with 2 strands of DNA, and each strand has one copy of the original DNA and one copy of newly constructed DNA. And remember, those are called our template strands for the original, and the non-template strand for the strand that was built. So we're going to kind of pick up where that left off. So we now have 2 complete copies of our DNA. Hopefully there are no changes. But just to kind of go back here, your enzymes, specifically your DNA polymerases, are not necessarily going to start at one end of the DNA strand. It starts simultaneously at various points on the strand. With the leading strand, the one strand that's going towards the replication fork, that particular one ends up with a very complete continuous strand of DNA, but it's still going to have different polymerases building it. It's just that they're going to be easily connected because they will run into the one before them. The other side, the lagging strand, has those Okazaki fragments. So, but then it needs the DNA ligase to go ahead and paste them together to be one. The whole point of having multiple enzymes working on the strand, especially on the leading strand, is to be more efficient, to get the DNA copied quicker. There are certain enzymes that will go back over. Sometimes it's a DNA polymerase, sometimes it's a different one, but generally it's a DNA polymerase enzyme is going to go back and just quickly proofread the strand to minimize the number of errors. So if there happens to be any change whatsoever in that DNA base sequence, so in the cytosine, guanine, adenine, and thymine sequences, then that is technically considered a mutation, even if it's only just one base that's been altered, either deleted, substituted, so maybe an additional base was inserted by accident. Any one of those would be considered a mutation because it's a change in the nucleotide sequence. Where this becomes very apparent or most impactful, you might say, is if it occurs early in the development of a fetus or an embryo, as they don't have as many cells as an adult human, much less a full-term infant. You still have significantly fewer cells. And since this is all based, since your replication is always based off of a template strand, if you have an error that's formed in a non-template strand, eventually that non-template strand will become a template strand when that DNA molecule goes through replication again. It then becomes a template strand, and then you're basically making a copy of that mutation. So if it occurs early enough, you can end up with more of those mutated strands than you would of the correct sequence of strands. So depending on the severity of the mutation, how many bases were affected and how early was it, that can lead to some significant physical or chemical, mental characteristics of that developing fetus. So we kind of talked about this yesterday about how replication can be associated with cancer, because that's essentially what cancer is just cells that are replicating uncontrollably. They haven't been, for whatever reason, they are not turning off. They don't know when to stop replicating. Your body naturally, when you have a scratch, knows when to stop. When when we replaced all of these skin cells, when we have replaced the cells that make up those blood vessels, your body knows to naturally, okay, we've sealed off that blood vessel. It's now complete again. We don't need to be replicated anymore. And so the genes turn off. The genes turn those proteins off, and so then replication stops. Cancer is when those genes don't turn off and we keep replicating, and you end up with these masses of cells, also known as tumors. So one of the big things that oncologists will use to fight cancer are chemotherapeutic drugs, oftentimes referred to as just chemo treatments. There are many different types of chemotherapeutic drugs. Essentially, they're all trying to do the same thing. They're trying to mimic the 4 different nucleotides found in your DNA, and they're trying to trick those cancer cells into stopping the replicating. They stop them at various points, depending on which type of cancer you have, and maybe where it's located, how far along are you. But sometimes they will try to stop the replication in a specific stage of mitosis. Sometimes they will try to stop it during the cell growth phase or something like that. So it just really depends. But basically what they're trying to do is they're trying to trick those cancer cells into thinking that these drugs are the DNA. And then they've altered that. The drugs have a different code, if you will, and that ceases the replication. Even though they look really similar to the DNA that's been coding for these cells to keep replicating, they have something in them that allows that replication to finally stop. At least we hope that it tricks those cancer cells. That's so that if it doesn't work, then at that point you would say that the cancer did not take up those chemotherapeutic drugs. So it didn't pick up and mistake the drugs, quote unquote, nucleotides for the cell's DNA. So what we've got is an experiment that occurred a long time ago, 1958. And this is where the idea of semi-conservative replication came up. So that, remember that idea that when you're done with your replication, you've got one original template strand and one non-template strand. So these two gentlemen, Meselson and Stahl, ran an experiment that supported this idea of semi-conservative replication. So they took a bacteria, E. coli, and they grew it in some agar that had nitrogen 15. Normally, nitrogen has an atomic mass of 14. They got nitrogen 15 by adding a neutron to the nitrogen. So that's why it's called a heavy isotope, because since you're adding a neutron to the nitrogen, you're obviously getting a larger mass. So that's what we call it, heavier. So this E. coli is grown in this quote unquote heavier version of nitrogen. The nitrogen is important because we have nitrogenous bases. So as the E. coli is going to replicate its DNA, it's going to use those nutrients in the agar to make more DNA and to grow and multiply into more cells. So since all they have is this heavy DNA, heavy nitrogen, nitrogen, not DNA, to use to make their DNA, they're going to have quote unquote heavier DNA, because each nitrogenous base is going to have more neutrons. On the flip side, you would say that bacteria grown with normal 14 nitrogen 14 are gonna be a little bit lighter. They're not gonna be heavier. Now the way they are able to separate these was kind of like how they were able to figure out back with the DNA that the DNA, not the protein coat, was what carried our genes. So they were able to, since this DNA is heavier, if you put it in a centrifuge, it's going to sink to the bottom or be lower in the test tube than bacteria that have been grown in the normal medium with the nitrogen 14. It's going to be a little bit more towards the top, the bacteria that would have been grown with a quote-unquote normal nitrogen medium. So that'll be important here in just a second. All right. So after they had allowed this E. coli to replicate and really grow a substantial colony infused with this nitrogen 15, think of it like a tracker almost, like a fluorescent tracker. It's kind of the same thing. They wanted to kind of track it. They then allowed the bacteria to grow in a normal growth media with nitrogen 14. So they knew approximately how long it took for the bacteria to replicate once. And so that's how they were able to monitor that it only reproduced once. So as these bacteria are reproducing, they are going to be building those strands of DNA. So you end up with one copy of DNA in each of the cells. And since they only have the quote unquote normal nitrogen, the nitrogen 14, we'll be able to track this now. So when they then put this generation of bacteria into the centrifuge, it actually popped up in between where the heavier nitrogen 15 bacteria were and the normal, quote unquote, bacteria that had the nitrogen 14. So it's kind of in the middle. So what this tells you is that it's kind of a hybrid, meaning it's got half of the heavy and half of the normal. They called it an intermediate density. So they were able to kind of track that. Then they allowed the DNA or the bacteria to reproduce again, also in the normal nitrogen 14 medium. And then they ended up with two different versions of density. So before, they had every single one was in that intermediate density. This time around, half of the bacteria, they had equal amounts. Half of it was in that intermediate version, and then the other half had the normal density again. Here's that picture, or a picture. You've got the original parent molecule. This particular picture has designated the heavier DNA as being darker. So this is your nitrogen 15 heavy DNA. It's allowed to replicate once. And as you can see, each particular strand has one of those heavy strands of the nitrogen 15. And then since these guys, this first generation right here, was allowed to replicate in the normal version of the nitrogen 14, it's going to have the quote unquote normal, or what they've designated as the lighter strand of DNA. So they're identical. So this is why they had the intermediate density. They weren't heavy like this one with two versions or two strands of the nitrogen 15, but they weren't light enough to have the two normal nitrogen 14s. Then they allowed them to reproduce again down here, like you can see, in the nitrogen 14 medium again. And then this particular strand right here became the template strand. So it was originally the non-template strand, but now it's the template strand over here. And since they're being allowed to reproduce in the normal nitrogen 14, it's going to also build that lighter strand also. So this complete molecule right here is going to be the lighter, complete nitrogen 14 density. But this guy over here, it still has the original strand of the nitrogen 15 right here. And since, again, you're still growing this in the nitrogen 14 medium, it's going to build a second strand, which is going to be that lighter version of the nitrogen 14. So of the 4 strands, 2 of them are at that nitrogen 14, 2 of them are in that intermediate, which is why they ended up with half of the DNA in that intermediate section and why half of them were in the nitrogen 14 or the lighter section. So this kind of explains how they were able to figure out that we don't completely take this original parent molecule, unzip it, build a whole new strand, and have half or have one complete original copy and then one complete newly created strand of DNA. Because if that had happened in the first generation, you would have had both versions. You should have had only, you should have had half of your DNA being in the heavy and half of it being in the lighter nitrogen 14. So that's what would have happened. That's what should have happened if we didn't have this semi-conservative replication. So if one strand wasn't passed on. So hopefully this makes some sense. I believe I've got a worksheet for you guys now to kind of complete and tie all of this together. So again, if you do have any questions, please let me know. Otherwise, this kind of wraps up the replication process.